energy reserves, the starch and whatnot, and put them into a usable form such as sugars. This particular part here is extremely important for things like brewing, right? Those of you who maybe have had the occasional beer, this is part of the process that is fundamental to your enjoyment of that beverage. Or things like whiskey. This is called malting, right? This is the conversion of starches into easily used forms of sugar and things like that is malting. And that's that whole sort of activation of enzymes and conversion of starch to sugars is part of the process that makes all of those things possible. Another thing that happens is there's a very strong increase in respiration that happens as the plant is starting to become physiologically active, as the seed is becoming physiologically active. Cell replication begins, the embryo gets enlarged, the seed coat bursts open, and the growing tip of the root, this is, it says the growing plant tip, it's actually the root, the radical, emerges out of it. Now at the bottom of the first page, you'll see a diagram, right? And I'd invite a class to, say, identify where germination actually stops then. And the rest of the thing is seedling establishment. Would anyone want to hazard a statement on the basis of this? No?
What was the question again?
Where germination is actually completed on that diagram. It's actually by the second, the second image there. The rest of this is all seedling germination or seedling establishment. So germination largely is in that invisible part, that just what happens in the before you actually see the tip of the plant. Now you don't have a copy of this. I didn't put this one on, but this is the endosperm of the seed. This is a corn kernel. I've got a bunch of different seeds that I'll let you have a look at. This is a corn kernel. This is all the endosperm, which is the energy reserves, the starch and whatnot. There's proteins and things in there. The cotyledon, the aileron is a protein layer. We have the actual germ in here, which is where the growing tips are. The two meristems are the growing tips. This is where most of the activity will begin when water comes into the seed, and it will start converting this material into usable sugars as germination begins. And there's that diagram. Now, I've done some little cassettes here. I'll pass them around. I'll start them in different ways. What they are sort of a plastic, well, an acrylic plastic sheet with some paper towel and some elastic bands. And I put some different types of seeds. There's radish, there's onion. I've got, so you can have a look at the different types of seeds. I've got some onion, alfalfa, radish, chia, mustard, cabbage, and some scarlet runner beans. So you can see these huge beans compared to these other ones. Yeah, sorry, it's gonna drop water on you if you pick it up and look at it. So I'll start these going around. I want everyone to sort of have a look at how these are done. Just have a sort of a quick look and pass it on. I'm not sure. Yeah, I'll start some of these going this way. This is just so that you can see what the sorry. Yeah, they were all started at the same time, so you'll see that some of them are going at very different rates compared to other ones. There's also one here where I've done them in little plastic bags instead of in the acrylic things. Right? You get the plastic bags. The thing about the plastic bags what I was trying to do was look at some different versions of sprouting the seeds. The plastic bag one is one that is a recommended technique for doing a seed bioassay. Now in this version here, what I've done is some seeds in paper towel that were just in water, some that were in 10 mils of I would have thought, a very mild salt solution. And you can see that there's maybe a little bit of activity, but nothing much there, and then absolutely nothing in the stronger salt solution. I was going to do some copper sulfate and some other things as well, but I just didn't get to it all. But the point of this is you can use the growth of the seeds, which remember I said was a very sensitive process, to evaluate the toxicity of solutions. And I'm in this version here, I'm trying it in the bag culture so that we don't have toxic solutions dripping out. These ones, although they work very well and they are nice and easy to see, they have the disadvantage of dropping solution, and so you'll see water dripping out. If you were using a toxic material, a pesticide, an oil, something like that, you would have a problem of dropping residues all over the place. Now, these are cassettes that I didn't do anything with, but for bigger seeds like beans, you would have to have a bigger container, right? So you would have to have something like that for soil or perlite or a lot of paper towel if you were going to use something for a bigger seed like that. Well, I did some radish. Those ones are radish. Yeah, so here's the actual, a range of different seeds that you can get from, this is from a health food store, Pico in town. You can see that the sproutable seeds are actually quite cheap. So we just had these in our fridge. We do sprouting and things for salads and things.
So, you just fun.
Yeah. Yeah, because it is fun. I made these things because these are really easy. This happened to be sitting in the back of our classroom down there. And although this is not set up for doing seeds, you could do seeds. If you wanted to assay, for example, contaminated soils, if you wanted to look at farm soils, look at issues of farm soil compaction or other aspects of behavior of soils, I see no reason why you couldn't use something like that. When I saw this, I thought, wow, that's cool. And basically, it's one of these, just on a bigger scale.
Where do you get these from?
I just had the acrylic sawn up. I bought some scrap for like $1 a sheet of offcuts of this, and the guy at the plastic store just cut them up for me. This was a store down in south of Morehouse.
I guess you could get them in hardware stores.
Probably not. You might have to use something like cassette cases, DVD cases, CD cases. You might be able to harvest some of that, or you could use some of the plastic from juice containers. If you did use, now this what I was using for the backing on these is just core fluid, right? So, and it produces a nice dark container, so you can look at things like phototropisms and things. You'll see a lot of these are bent strongly to one side, right? I don't have any markers for this, but are there markers here somewhere? You can see. Oh, cool! You carry these with you? Okay, you'll see that some of them, the plant is, they're all doing this sort of thing. That's because I had these growing on my refrigerator, and the light is over here, right? So the seed would be in here, there'd be roots forming this way, There are so many things you can do with this. I get really kind of into this.